Okay, good morning. My name is Ethan, and today my topic is about the trophic relationship of the hydrothermal vent and the non-vent community in the upper sublittoral and the upper bathyal zones of Kueishan Island, Taiwan. A combined morphology and gas content analysis and stable isotope approach. And this in the Journal of the Marine Biology. And first, we know the Milk Sea is a hotspot in the Kueishan, but we know about the Yushih that has have a word for that is very hot, very acid, and there's full of sulfur. And here's a okay, I think it's broken. Okay.
I think it's
Working?
You have to wait.
No. Okay. I'm out of time. Okay. So the video I want to show that is there's a crab like this, and it's this kind of crab, and we call it in Chinese we call 乌龟怪方蟹, and and in my presentation I will call it the turtle crab because it's better to call it. And turtle crab it will live in such a dangerous environment. And in this paper, it said what turtle crab is eating. It's eating the xeno. It's the zooplankton killed by the vent plants. And in this paper, it also said they are highly mobile. And we are interested, scientists are interested about what others creature in this kind of the dangerous environment. So what is special in the Kueishan vent? And the Kueishan vent, we also know is about the chemosynthetic. And but the special is in this, because it is the costal one. So it can come energy from the sun. And it might be a mixed photosynthesis chemosynthetic system. And we have to examine whether it is kind of system. And another thing is that we want to know this kind of food web here. So how do we to construct our food web? We use the stable isotope tracer. And there's a kind of word that you are what you eat. And what is stable isotope? Stable isotope is about they are not radial and they are steady in nature. And what's called the stable isotope different is called isofractional. The isofractional make out the constant may be the different weight of the elements. And there's two parameters that we can cause the food web. One is delta 13 carbonate and one is delta 15 nitrogen. And if the predator and the prey they will have the almost same carbon, and the predator will also have the higher nitrogen. So we can build a food chain. Okay, so this is our aim. So we want to know the habitat trophic structure, whether it's the photosynthesis, chemosynthesis, or both energy path. And one is the ontogenic dietary shift on the turtle crab. What is ontogenic? It means it will change its food habitat. So another thing is that we want to know the crab transfer energy. That because I have mentioned that the crab is highly mobile. So whether it will go other not the vent zone and to transfer the energy. Okay. This is our study place in the Kueishan. And the square is about the, I say the sublittoral is mean the coastal. And this triangle is mean the better one, is more deeper one. And the circle one is might be no vent. And how do we to detect the bathyal have the whether have the hydrothermal vent? And we use this technique like high frequency underwater sonar. So this might be a plumb here into a bubble, and here might be a vent. And we also capture the fauna here by scuba diving, and here some deep water. We use the trawler here, and we have 3 methods to examine our aim. The morphology of some crabs, the crabs or the creature, the gut content, and the isotope. And the morphology will examine the chela and the pyrite parts here. Chela is this, and pyrite part is this. And to classify its feeding modes. And isotope we will use this, iron mouse. Okay, so our results, and this the author, he wrote a lot of beautiful picture, and I'm not going to talk details. And my important presentation is here, isotope analysis. The x-axis is the delta 13 carbonate, and the y-axis the delta 15 nitrogen. And you can see different icons mean the different species, and it is on the upper sublittoral veins. It means the coastal one. And first, we have to see, because there are little overlap, they are not very similar. So we cannot find very much similar relation about the trophic. And the second thing is I want to say about this dot line. And we can say about smaller than the -22, the carbon sources might be caused from the CBB cycles. CBB cycle is also called the Kelvin cycle. And the right one is the mixed carbon source. And the first, I want to say about the first creature is about not the crab, it's the biofilm on the crab. It's here. And he have the lower nitrogen. So we might think this the primary base of trophic. And this another creature. These two is the alga, green alga and the red alga. And because it is in the low carbon and also low the nitrogen, so we will say it's a primary producer. And because there are no overlap with other organisms, so we will say there is not predator. Okay, and this is the red and black animals. This, the orange one. And because they are very close to the zooplankton, this one, so we have to say maybe there's a close trophic relationship, but to the crabs here, to the zooplankton, they are too far, so we don't have say they have strong relation. Okay, this the place on the upper bathyal veins. And this kind of at least muscle, and it said it has a low data, the carbon value. So, but it suggests that there have some chemoautotroph bacteria in here, in their body. And another thing I want to introduce is the this kind of mudship. Because their variation is very big, so we are call it the generalist omnivorous. This means they can eat many kinds of food, like white food resource and bacteria. Yeah. And this one, the vein shape, the orange. And this with some the cutting edge were very not too sharp, so we call not a predator, but some scavenger or the deposit feeder, and might be suggest that it feeding that the living microorganism or the myofauna. And to this, compare this. This is the mussel to the shrimp, and there's no direct relationship. Another one is the hermit crab. This hermit crab, and they have the this one, setae, and there might be have some bacteria on it. And another thing is that they have the highest value of the carbon and the nitrogen, and close to this non-vein fauna. So we are also say it about the generalist omnivore, and have a lot of food resource. Okay, and this to the carapace length, carapace. And the x is carapace length, and the y is the isotope value. And only in the delta 13 carbon have a relationship. So we come out that, oh no, this is not, so this is a result. And we have to discussion the crab, the turtle crab. And the turtle crab here, in different value, isotope value, we can come out there are different food sources. And in the this I just said about the length, and we can suggest that there's an ontogenic shift because in they're small, they cannot eat too big carbon. They only can eat the none of the carbon, the none of the 13 carbon. Yeah, so there might be an ontogenic shift. Okay, and I'm going to say about the mobility. And although there's a turtle crab collect in a non-violent environment, but the isotopes show the vary in the upper band here. And in other resources, protein expression can rapidly acclimate different environment. But there's need further studies. And it says we have to know some submarine machine to help this kind of study. So in our conclusion, the heavy trophic structures on this costal vent, we need more investigation. They're not just classify. And of the bathyal vent, is a chemo-autotroph based system. We can see many bacteria. And is this ontogenic dietary shift? Yes, there's in the upper bathyal vent. And in the transfer in injury, there needs some more studies. And thanks for your attention.
Can you explain again isotope analysis?
What's the question?
If you want to know why is isotope analyzed. Here. Okay. Because we want to construct a food web, and we use the stable isotope to be a tracer. And because there might be some difference about the, or be the sum of the same of the isotope, so we can come out this kind of this is the perfect figure we want to know, we want to get, but in our research there's not so beautiful. Yeah.
Could you briefly explain your discussion? Because it's too much information.
Okay, now it's just a conclusion, I think. So this research want to know three things. And the first thing is want to know whether this kind of the food web is what is its food resource. It is about the chemical autotroph of the photosynthesis. And our research come out is the upper benzol is the chemical autotroph. And on these two things, we want to know the turtle crab, whether have this kind of will match their hypothesis. But in this, the data reshift, they say yes about the band event. But on the transfer energy, they say it needs more studies. Yeah, thank you.
In result, you use the orange test. I don't know what it mean, and yeah. The dash, orange dash.
Okay.
What's the question?
She wants to know this dash, what this means. It is a line divides the carbon value, and you can see in the low value, it said from other research, it come up that it is a reaction that we can say about is come from the Kelvin cycle. Yeah.
Could you back to materials? Could you explain again about the solar pictures?
Solar picture.
Solar, the lower.
This one.
Yes.
Okay. This means how did they detect the hydrothermal vent in the seafloor? Because this is the simple way. In the earliest way, they find the hydrothermal vent by this sonar, and you can see here, it might be some bubbles and the plumb, and here there's nothing, no structure. So there's no hydrothermal vent there. Yeah.
What is the purpose of the research?
It's not a good question. Okay. I think it's two. No, it's two. Yeah. First is to want to know the habitat structure, and the second they want to know this kind of crab whether it makes their hypothesis, and this is their purpose.
Why do the delta carbon 13 and nitrogen 15 represent to the food source and food web?
Okay. Because we know there are the stable isotope, so they cannot change very extremely by any other factor. So these very stable sources. So if I eat the sources, then I will become what I am. Yeah, and the nitrogen. Why the nitrogen can be the food chain? The food chain is because the nitrogen. They will eat more nitrogen in their body, so maybe the nitrogen will be more higher than their prey. Yeah.
Regarding your talk, first of all, he at the beginning he said he would not call these 乌龟怪螃蟹, this difficult Latin, and he used turtle crab, which is very easy to remember, which is good. And then there are a few pronunciation problems. I think most of us will say hydrothermal plume instead of plum, because plum is a kind of fruit. Yeah. And then this reads fractionation. And then he has actually a very clear end of purpose. However, based on what I heard today, I think, although I know this might be the whole purposes the authors wanted to achieve, I would say you probably take away data and discussion of this topic. Because this, after listening to your talk, this one leaves no impression at all. I don't remember what you said. This one. I have stronger impression of this. And because you spoke a little bit over time, so you probably need to take away part of your results and focus on the other more impressive results. And then when it comes to your results slides, it's I would say if you are not going to read all this, you can save our brain by remove some of the text. Because I was thinking, oh wow, you're going to read all these. Wow. So if you are not going to do that, you can make your slides cleaner by removing unnecessary text here. Otherwise, I don't know where you are talking about. I was looking around. Then the same actually applies here. Do you need to look at this in order to read your line? I don't think so, right? You remember your line in your head. You do? But I think you can do better next time. Because when you have this, it's really like, and you have so many keywords, it's really like I'm really looking around and see what is the key message. It's a bit distracting, then I'm not listening to you. If you can just print keywords, just keywords, so it's easy to read and easier to follow instead of these messy slides, difficult to follow. And the same actually applies here and there. That's one of the reasons that at some point I'm a little bit lost because there are so much information I can't follow. So next time, please condense your text to show just keywords or key message. And then also in a slides like this, these are actually very small font. Maybe you are aware of. These are really small. If you condense your text, you can enlarge the keywords. It's again make your slides much more readable. All right. And then here there are actually a lot of information. So again, like next time, I suggest that you remove some of your results and explain the figure slowly and more clearer. For example, here you are talking about something like a satay, satay or something. I was looking around where is the keywords, and I don't know. Then I got lost. So this kind of thing should be improved next time. Yeah, and then this to save you time, the discussion should be also condensed. Like I said, energy transfer should be avoided, and then this one can be removed as well. And then so that you can have you can make sure you finish your talk in 12 minutes and complete your finish your conclusion. All right? Okay, thank you, Ethan.